Hello, good evening, what's up, and welcome back to another video in our 1001 MSc online tutorial series. My name is Brock Grant, and today we're going to be covering an introduction to biological molecules. So today we're going to be covering a few different topics. We're going to be looking at hydrogen bonding. We're going to look at some biological reactions and biological molecules that we see inside of living cells and living animals, including ourselves, and trying to sort of establish a link between what you guys will be learning in CTR and how these cells work, how we as a system work, and related to a bit of chemistry. So a few prerequisites for this module is I highly recommend that you watch the Module 5 Video 1, Chemical Bonds and Molecular Geometry, and Module 3 Video 1, Atomic Theory. Now, in regards to these videos, if you make sure your annotations are turned on, so in the YouTube video screen, there's a little button that looks like a gear lever. If you make sure annotations are on and you click on one of these here, it will automatically fast forward you to that section of the video. So just a quick reminder again of our video symbols. So whenever you see our little owl here, it means that this is going to be a very difficult topic, and it may take a little bit more time and attention for you to get your head around it. Whereas if we see our little oops symbol, that means it's a student trap. It means that a lot of students do make mistakes in that particular section, and to keep your eye out for it. So let us talk now about hydrogen bonding. So a hydrogen bond is formed when you have a hydrogen atom that is already bound to something that is electronegative, like oxygen or fluorine or nitrogen, and it's attracted towards another electronegative ion. Now, a good way to sort of think about hydrogen bonding is to not picture it in your mind as a bond. Now, I know that may be a bit confusing. Now, the reason for this is that when we were talking about covalent bonding and ionic bonding, it involved the movement of electrons. So I want you to think instead, when you see the term hydrogen bonding, I want you to think hydrogen attraction. Okay? Because they're not formally binding together. It's not a formal exchange of electrons. It is just an attraction, like we find with magnets when we have positive to negative. So a good example of where we see hydrogen bonding in play is when we look at water. So if we have a look at water, now in our previous video, we did look a lot at polarity, at dipoles, at all sorts of stuff like that. So we know from our previous video that oxygen is delta negative and hydrogen is delta positive. Now, because of that, if I was to have another water molecule down here, what would happen is these delta positive hydrogens would be attracted to the delta negative portion of the oxygen with this other water molecule. So again, it all comes down to positives and negatives attract. So because this is a positive, this is a negative, they're going to want to come together. And it is for this reason that we form ice, because we have this hydrogen bonding. We also see hydrogen bonding in our DNA, and it's what holds our DNA together. And we see it a lot in a lot of other different molecules, and it can affect a variety of different properties such as boiling point and melting point and so on. So it's just very important that you understand hydrogen bonding and that you don't get confused between thinking that it actually exchanges hydrogens. So big takeaway message here is that it is a attraction between a delta positive and a delta negative. And it is a very, very weak attraction. It is very easy to break a hydrogen bond. Now let us look at some biological reactions. So these reactions, this is how we would sort of communicate them. This is how we would write them. And it really paints you a story as to what these elements are doing and what's happening. So we have two very important terms that we use. We identify them as either the reactants or the products. So reactants are our initial elements. So the reactants will react and they will form our product. So if we have a look at a synthesis reaction or anabolism, so this means to build, to construct. So if we have a look at 2H2 and O2, So these two here are our reactants. So I'll just draw an R to represent reactants. Now, as the hydrogens and the oxygens react, now that is signified by this arrow. So think of the arrow as something is happening. So we have our hydrogens, we have our oxygens, something happens, so we look away, that's the arrow, so something happens, and when we look back, we have water. And this is what this reaction is telling us, which means that the product is H2O. So this is an anabolic reaction because it is, you have hydrogen and you have oxygen, and they're combining to build water. Whereas if we look at a catabolism or catabolic reaction, this is where it decomposes. So we have one thing that breaks down into more than one thing. So if we look here, we've got H2O2. So these again are our reactants. And they react. So we've got our hydrogen peroxide that breaks down. So something happens, and we're not sure what. So we've got H2O2, something happened. And then as we look back, we can see we have H2O and O2 as our products. So because we started off with one reactant and more than one product, that is a decomposition reaction. So we also have exchange reactions. So exchange reactions are essentially a combination of synthesis and decomposition reactions. So the best way to sort of picture this is imagine that you've got two couples at a dance. So we've got couple A and B, and we've got couple C and D. And what they do is midway through the dance, they swap partners. So A goes with D and B goes with C. So they sort of switch. So the reason why this is sort of both a decomposition and a synthesis reaction is because in this reaction, they break apart, but they rebuild in a slightly different way. Now, let us look at some more important exchange reactions. We looked at what is called hydrolysis. Now, this is important to understand, but another giveaway too is in the name. So if we look at hydrolysis, so hydro meaning water, and lysis simply means to split, to break apart. This is why it's called a lysosome. Okay, so it's the same principle. So a hydrolysis reaction, or hydrolysis, is just simply the splitting or breaking of something by water. And that is what we see over here. So if we have our polymer and we add water into the mix, this water can actually break apart, can actually split this little bond here, holding them together, meaning that we're going to have this polymer here going off in its own merry way, and we've got our monomer over here. And they both split and they move apart into their different areas. So that's a hydrolysis reaction. Now, we also have condensation. So condensation is essentially when you're using water to build things together again. So hydrolysis and condensation are sort of opposites. Now, we have what are called reversible reactions. Now, although we do have some reactions that move in one direction, so that means that we have our reactants, we make them react, and we have our products. And so far throughout this course, that has been, we have just assumed that all of these reactions have been like this. However, unfortunately, in real life, things just aren't that simple. What we do have what are called reversible reactions. Now, these reversible reactions are indicated by a double arrow. So before we've shown single arrows, so instead of it looking like this, it will look like this. And this basically means that depending on the environment, depending on what it's mixed with, this reaction can go both ways. So it could either A, go from AB and split to form A plus B, but depending on the circumstances, a lot of environmental conditions or concentration, the A plus B can combine again to form our initial reactant. So just because we have one reaction and it forms a product, it doesn't mean that that reaction can't move the other way and take that product, recombine, and form our initial reactant again. So this overall topic is called equilibrium, and it's something that will be taught more in depth next semester. Now, it should also be noted, as I said before, that there are a lot of environmental conditions that can affect the movement of a chemical reaction, whether it's going to favor more the products or whether it's going to favor the reactants. So this, when we have something that's going to influence or push which direction it's going to go, we will write that above the arrows. So, for example, as we can see in this reaction over here, we have AB. Now, if we were to add water to AB, it would then encourage the creation of A plus B. But if we had our A plus B and we wanted to push it the other direction, we wanted to turn it into its original reactant and combine them again, this is where we just add heat. So by adding heat, it will encourage these chemicals to bind together again and form our initial product, which is AB. Now, before we get too caught up in looking at different sort of biological molecules, I really want to cover a few of our definitions, because this is where a lot of students get tripped up, especially in an exam situation, because they want to, like, you'll go through, read the question carefully, but it makes it very difficult if you can't understand what the question is asking you. So, a couple of things here. The first one is macromolecule. So if you see something referred to being a macromolecule, so macro means large, opposite to micro, and molecule obviously means a molecule. So a polymer, so poly just means multiple. So all it means is that it's a large chain of subunits. So we're going to cover that in a moment when we look at carbohydrates. A monomer is just simply a molecule that combines with others of the same kind. So if we have a monomer, we can have multiple monomers join itself together to form a polymer. Okay? If you see the term saccharide, saccharide is a very chemistry fancy term for saying sugar. So something referred to as saccharide is a sugar. So if we have a polysaccharide, okay, a polysaccharide is a long chain of sugars. And the last one is peptide. So peptide is just chemistry speak for a chain of amino acids. So amino acids are just the monomer for proteins. Now, something too I should mention, just going back to our saccharides, if you have a chemical that has a suffix or ends in the word ose. So glucose, fructose, maltose. This ose represents a sugar. So that's a little sort of thing to add on to your suffix vocabulary, and that if you see the word ose at the end, it is a sugar. So now that we have our terminology done and dusted, let us now move on to the different types of biological molecules that we will see in our human body. So the first one that we're going to talk about is carbohydrates. So a carbohydrate is essentially a group of molecules that include like sugars and starches, but essentially a carbohydrate is just a long chain of sugars. That's it. That's all it is. So inside the human body, its primary function is a source of chemical energy. So it basically allows us to store sugars away that we're not using at present, and use them as a fuel source later on. So if you've been in the lab for 3 hours, you haven't eaten in a while, your body needs energy. It will then rely on these carbohydrate sources in order to fuel yourself and get yourself by until the end of the lab when you can have a feed. Now, carbohydrates are also used in structures. They're also used to sort of build and sort of support things. So we see this in DNA. So it's deoxyribonucleic acid. So ribose is a sugar that is seen inside your DNA. Now, in general, carbohydrates are classified according to their size. So if you see a monosaccharide simply refers to one sugar. So mono meaning one and saccharide meaning sugar. So these are things like glucose and fructose. And it is just one sugar molecule by itself. So it's not joined to any other sugars, and it's about as broken down as we can get it. Now, a disaccharide, so di meaning two, obviously, as you may have gathered now, it is just simply two sugar molecules joined together. So this is where we see things like sucrose, like our normal sort of table sugar, and lactose. So if you're lactose intolerant, this is what you struggle to break apart. So, and that is just simply the combination of glucose and galactose. And lastly, we have a polysaccharide. So poly meaning multiple. So it's multiple sugars. And this is where we see glycogen, which is the primary form of storage of saccharides, of carbohydrates, that is. So when we want to store our carbohydrates for a long period of time, we form it into glycogen. Now it is also it is also called cellulose. That's another polysaccharide. Now we see those primarily in plants inside the cell wall, and that's what sort of encompasses that cell and provides it a lot of protection. Now, in regards to carbohydrates as well, we see our condensation and hydrolysis reactions being quite common with these. This is the main method that we use to break these carbohydrates apart or build them together. Now, a bond that holds two sugars together is called a glycosidic bond. And that's very important. You must know that. Okay? You have to know that it is this type of bond that is responsible for holding two saccharide units together in a disaccharide or a polysaccharide. So again, this is what we can see here. So if we have our glucose and we have our fructose, we can see that we have our two hydroxyl groups. So what will happen here is that one of these hydroxyl groups will bind to one of the hydrogens from the other sugar. That will then release water, and that will then form this bond. So this bond here is called a glycosidic bond because it is this part here that is responsible for holding these two sugar molecules together. Now, a reverse reaction is also true. So if we see here, if we were to hydrolyze this and we break this apart by putting water in there, it will then split these two sugar molecules apart and leave them in their two individual units, which is what we see over here. So glycogen is the most prominent polysaccharide in the human body, and we use this again as storage of glucose molecules. So glucose is the primary energy molecule of our body. We use that the most to extract the most amount of energy to fuel us. So what we do is, as an energy store, we get a lot of these glucose molecules and we bind them together. So we have a long chain of glucose and we stash them away for a rainy day. So then once we need a quick jab of energy, we can grab one of these glycogen molecules and start snapping off the glucose molecules to use them for energy. So here's what we can see here. So this is our glycogen. And we can see we just have lots and lots of these small glucose molecules just being chained together. So when we want energy, we will simply hydrolyze this and we'll just start splitting these bonds here. So we'll break all of these bonds apart, and then we will then use those glucose molecules to provide energy to our bodies.